improve gene-edited animals for genetic improvement or food use. Our first speaker will be Professor Wong from Northwest A&F. He currently is leading their lab mission to apply genome editing for genetic improvement of small ruminants that promotes sustainability, animal health, and rapid increases in meat and fiber production. So really their work is geared towards making animals for smallholders. And if you didn't know, there's 1.5 billion smallholder farmers in the world, so very important application. His main interest is in cashmere goats and improving their fiber, which improves the profit for the smallholder. And then also being able to use indigenous sheep breeds and improve them rapidly for their muscling and their wool production and climate change and parasite resistance. And they've produced thousands of animals, and I'm showing just a few of them here on the screen. The second speaker is going to be Kristin Whitworth. She's a professor at the University of Missouri and the Associate Director for Research Activities at the National Swine Research Center. This is an NIH-funded center established to develop swine models for biomedical research community. And Kristin's research focuses on efforts to use gene editing tools such as CRISPR-Cas9 to create pig models for both disease resistance and biomedicine. And if you didn't know, the majority of the world's pigs are in China right now, and they're undergoing their own pandemic, which is shown in this map at the bottom, with African swine fever virus, something that border controls in countries that aren't are very concerned about. So this research topic is very timely, not only relative to livestock, but also to the pandemic we're currently undergoing with COVID-19. And our last speaker will be Professor Walt Lowe. He's in the Department of Neurosurgery and Stem Cell Institute at the University of Minnesota. His research is focused on the development of stem cell and gene engineering technologies for the repair of the nervous system. His recent work involves the generation of exogenic organs and cells using blastocyst complementation in gene-edited embryos with the ultimate goal of producing human organs and authentic human cells for transplantation and regenerative medicine. Both Professor Whitworth and Professor Lowe will have breakout sessions, and I'll add that the first talk is pre-recorded, and if you have any questions for that, they'll be mounted on the discussion board during this session. So with that, I think you did point out some very important things, and that is that the actual natural mutation rate is much, much greater than any off-targets caused by the editing tools, as long as you're doing your designs appropriately. So a good take-home message there. Now I'm gonna turn over the presentation to Kristin Whitworth.
Well, thank you for the invitation to talk today. I appreciate that, and and I think this format's going pretty well. So thank you again, and thank you for the introduction, Tad. I'm Kristin Whitworth. I'm at the University of Missouri, and all of this work was done in collaboration with Dr. Kevin Wells and Dr. Randy Prather, and it's my pleasure to present that. As was in the introduction, I'm the Associate Director of the NSRC, and most of this work was done, I've been in that position for 2 years, and most of this work was done when I was a research scientist at the University of Missouri. And to give an overview of what I will be talking about today, there's 4 different topics. I'll touch on the porcine reproductive and respiratory syndrome, the PRRS challenges, and the CD163 knockout pigs. I will then talk about the coronavirus challenges. I used to enjoy talking about coronaviruses, and I've kind of recently decreased that enjoyment, but we'll be talking about the TGV, PDV, and the Delta coronavirus, and how those ANPEP knockout pigs were challenged with coronavirus. I'll talk about a little bit about African swine fever. Most of this work was done at the Roslin Institute, but I do have a few slides and about some of one of their challenges. And then lastly, I'll touch on the influenza. So what is PRRS? I know we have a wide group of people represented today. So it used to be called blue-eared pig disease. PRRS is a devastating disease. It first emerged in the United States in 1987, and then later in Europe in 1990. By 2002, there were 28 North American type 2 PRRS viruses identified in Korea. By 2006, this disease emerged in China as a very severe form of the disease, the highly pathogenic PRRS or HP PRRS, which decimated the pig herds in China. PRRS replicates in macrophages. It causes respiratory disease in young pigs and reproductive disease in mature boars and sows. I took out some of the pictures that shows what PRRS looks like, but when you walk into a barn after an outbreak of this disease, there's abortions, dead pigs. It's a miserable disease, and to date there's been some work on an effective vaccine, but it has not prevented this disease from decimating pig herds throughout the world. There's a huge economic cost to this disease. It's estimated that $660 million are lost in North America annually. Further losses in Europe, this translates to about $6 million of losses to the pig industry every day. And this does not include losses in Asia, emotional losses, stress, and just the overall impact on animal well-being. So we wanted to make a pig that was resistant to the PRRS virus, and we did that via gene editing. So to start out, we looked at just some of the infectivity models, and this was an infectivity model for PRRS that was presented by Van Breeden in 2010, where this virus comes in, it has initial binding with heparin sulfate, it's then internalized by Siglec-1, and then thought to be uncoated by the CD163. So when we started this project, we looked specifically at knocking out Siglec-1. And this was done before the CRISPR and the gene editing tools were available. So it took quite some time to make these pigs. And when they were challenged and this work was published in 2013, they were not resistant to the PRRS virus. So at that point, we were already working on knocking out CD163 using gene editing tools with CRISPR-Cas9. And so for those of you that are not familiar with making gene edited pigs, we introduced the CRISPR-Cas9 system into fetal fibroblast cell lines, select for the edit of interest, and then we clone that pig. We can also now directly inject the CRISPR-Cas9 system into the pig one cell stage zygote. And this is just a picture of what those embryos look like and the machines that are used to do that, the micromanipulation microscopes. Those embryos are then transferred back into a pig, as you can see here, and then those pigs deliver their piglets. And so for the first time, when we did this, knocking out CD163, we had 4 different piglets born, and all 4 were edited with 100% biallelic edits. So, but I think the important question is are the CRISPR-Cas9 CD163 pigs edited pigs resistant to the PRRS virus? So those pigs, those founder pigs, were then bred, and we created a litter of CD163 knockout pigs that were then transported to Kansas State University for a challenge. And we did not want to bring the PRRS virus into our University of Missouri facilities and risk infecting some of our biomedical models, but Kansas State had the facilities that were appropriate for that. So they were transported to Kansas. And this is just a summary slide of all of the different CD163 challenges. We started off with the North American type 2 isolate. The young growing pigs were challenged, and those pigs were resistant to the PRRS virus. I'll talk a little bit about how that was measured below. We then challenged the pigs, a different set of pigs with the type 1 European isolate on young growing pigs. Those additionally were resistant. A group from the Roslin Institute made a set of pigs that had the domain, the PRRS binding domain deleted, that left the rest of the CD163 molecule intact, and those pigs were challenged and were also resistant. We went on to take CD163 knockout pigs, get them pregnant with wild type fetuses, and we were able to show that that the CD163 knockout pig protected the wild type fetuses from PRRS infectivity. And then this work was later repeated in China, and CD163 knockout pigs were challenged with the highly pathogenic PRRS and were also resistant. And so how that was measured is below. Whoops, let me go back. There was no fever. They were looked at clinical signs. There were no fever. There was no respiratory distress. There were no abortions in the pregnant pigs. No presence of viremia in the blood, no antibody response, no lung pathology, and no identifiable placental transfer. There's lots of different PRRS isolates, as I mentioned before, and we also did an in vitro panel of 15 different isolates, and that also showed no ability to infect the CD163 knockout macrophages. So at that point, we were confident that CD163 is the PRRS viral gatekeeper for that. But then there's also, there were many other swine viruses that we wanted to look at, one being the coronaviruses. There's multiple coronaviruses that infect pigs, the first being the TGEV, which is transmissible gastroenteritis virus, and then PEDV. PEDV, there was an outbreak in the United States in 2013 that resulted in the death of about 10% of the pigs in the US pig population. So both PEDV and TGEV cause malabsorptive diarrhea and dehydration and cause the destruction of the infected enterocytes and 100% mortality in young pigs. Another coronavirus is the porcine respiratory coronavirus. This is one that's a less severe form of the disease, and when the pigs get this disease, they actually get they get antibodies that neutralize TGEV. So a lot of pigs that if they've been exposed to PRCV, they don't get TGEV, but that's not the same case for PEDV. There's also the porcine Delta coronavirus. This one is concerning because there's some host switching involved. This was reported in China in 2002, the United States in 2014, and it also causes diarrhea in pigs. So just a little bit about the coronaviruses that we're looking at. There's 4 different genera of coronaviruses, alpha coronaviruses. The 2 challenges I was talking about, TGEV and PEDV, were alpha coronaviruses. Our least favorite coronavirus right now, the COVID-19, is a beta coronavirus. There's gamma coronaviruses, are common in avian species, and then the Delta coronaviruses. So we targeted ANPEP, which is a type 2 membrane metalloprotease. It's responsible for removing the N-terminal amino acids during digestion. It's highly expressed in enterocytes, and it's thought that the binding domain is between amino acids 717 and 813 in the ANPEP protein. So we made a set using CRISPR-Cas9. We made a group of ANPEP edited knockout pigs. We then bred those pigs and then had those pigs available for various challenges. We did find that after the in vivo challenge of these pigs, they were not resistant to PEDV, but they were resistant to TGEV. We then went on to challenge, those were both the alpha coronaviruses. We wanted to check a different category of coronaviruses, the Delta coronaviruses, and they were not resistant to the Delta coronavirus either. So after those 3 challenges, it was identified that ANPEP is the entry mediator for TGEV, but PEDV and the Delta coronavirus, they may use ANPEP, but there's clearly another molecule involved. And this is just a picture of what, I'm presenting a lot of information, but not a lot of data. And so I wanted to squeeze a little bit in there, but as you can see, along the bottom, I don't see my cursor now, but there's the open circles represent our ANPEP knockout pigs. And then this is just a PCR, a real-time PCR that detects virion. And as those pigs are infected with TGEV, the black circles, the wild type pigs, they become positive by day 3. And if you look at day 3, 6, and day 7, our knockout pigs never have the presence of TGEV viremia. And along the bottom, you can see some immunohistochemistry that we did not see TGEV localization or any immunoreactivity in the ileum of these pigs. So the conclusion for ANPEP, the absence of ANPEP did not profoundly affect PEDV or Delta coronavirus infection. The absence of ANPEP did prevent TGEV infection. And so again, it was determined that ANPEP is the intermediator for TGEV. So I'll briefly talk about African swine fever. African swine fever is a highly contagious, often fatal disease that causes hemorrhaging in the infected pigs, and there's no effective vaccine. China, most of Asia had a huge outbreak this past year, and it was devastating as the only cure is to euthanize the pigs and to get rid of them. Interesting, unlike domesticated breeds of pigs, warthogs do not die of African swine fever infection. And the difference between the pigs and domesticated pigs and warthogs is thought to be mediated by the NF-kappa beta motif in a gene, REL-A gene. And so REL-A is a subunit of a transcription factor that regulates immune response. So this is a couple of papers that were reported by the Roslin Institute where they looked to make a 2 and 3 amino acid changes in the REL-A gene to make the domesticated pig's REL-A gene more similar to the warthog ortholog. So they created these pigs. They either, like I said, contained either 3 or 2 amino acid substitutions in this REL-A protein. They were then challenged with African swine fever, and this work was published this year. The domesticated pigs with the 3 amino acid substitution did have a delay in African swine fever clinical onset, but they were not fully resistant to the virus. So the researchers there at the Roslin Institute concluded that there's including warthog traits within domesticated swine could potentially decrease the severity of African swine fever in our domesticated pig herds. There was some in vitro evidence that suggested the CD163 knockout pigs may be resistant to African swine fever. Those pigs were challenged in 2017, and they were not resistant to the African swine fever. So I also wanted to point that out. It seemed to be very specific to the PRRS virus. And lastly, I want to talk about swine influenza. This is a zoonotic virus, so it causes respiratory disease in both pigs and humans. How the influenza virus becomes active is there's a host transmembrane serine protease, TMPRSS2, that's been shown to activate influenza by cleaving the hemagglutinin and causing infectivity. We created a set of pigs at the University of Missouri in collaboration with Kansas State University, where we used CRISPR-Cas9 to create biallelic edits in the TMPRSS2 gene, and then determine if they were resistant to influenza. But to date, that viral challenge has not been completed, so we have no conclusions, but the pigs were created. So just in conclusion about these studies, I can go into more details in the breakout session, but for the PRRS virus, which is the SIGLEC1 knockout pigs were not resistant to PRRS virus. The CD163 knockout genes and the CD163 knockout pigs and the CD163 exon 7 deletion pigs were fully resistant to PRRS. A CD163 knockout sow can protect wild-type fetuses from PRRS. For coronaviruses and PEP, knockout pigs are resistant to TGV, but not PDV and Delta coronavirus. African swine fever, REL-A edited pigs were not resistant to African swine fever, but the 3 amino acid change in REL-A protein did alter infectivity onset. For influenza, pigs have been created, but those have the TMPRSS2, but they have not been challenged. And so with that, I just wanted to acknowledge the group of people, the large group of people that helped with this work: Randy Prather, Kevin Wells, the entire staff of pig management team, our group IVF and cloning team, Kansas State University, Bob Rowland, who is now at the University of Illinois, who participated in these challenge experiments, and then Genus funded the challenge studies for the work that the University of Missouri presented. And with that, I can take any questions at the breakout session after the next talk. Thank you.
Okay, so today what I'd like to do is to provide an update on the science of generating organs in chimeric animals and review some of the policies that are actually impeding progress in terms of generating human animal chimeras, and then talk about a survey of public attitudes that we hope here will provide feedback to sort of reform the current policies that the NIH has. So first, let me disclose that I have interest in these companies here and also equity in these companies. So when Jamie Thompson first discovered and characterized human embryonic stem cells, there was a great deal of excitement, and that with the possibility of actually generating human organs and cells for transplantation and regenerative medicine. And this was followed by the development of these inducible pluripotent stem cells, or iPS cells, by Shinya Yamanaka and his colleagues, where they were able to take transcription factors that were associated with ES cells and introduce them into somatic cells to generate these iPS cells. Then following that, these cells could then be differentiated into a variety of somatic cells and potentially generating, organs. So with this strategy, then, we have a lot of groups that have been trying to take iPS cells and to differentiate them into mesoderm, such as cardiac cells and smooth muscle, endoderm, such as lung cells and also pancreatic cells, and even ectoderm, such as neuronal cells. But in spite of the ability now to really generate a vast variety of cell types, the ability to generate whole organs has yet to be sort of successful. And so why is that? And so groups have now started thinking about really utilizing the embryo, the developing embryo, as an incubator for generating whole organs. And so the strategy is that, we know that the endoderm and ectoderm and mesoderm will generate these various organs, but transcription factors and key genes are really critical for developing these organs, could potentially be knocked out so that it creates a niche within the embryo where stem cells could be introduced and then develop into those particular organs. And so this was a strategy that was developed by Hiro Nakaguchi and his group in Japan. They were interested in developing a pancreas for isolating islet cells for treating diabetes. And so they targeted PDX1 as a gene. And so this knockout of this gene in mice, for example, caused these animals not to develop a pancreas. And so they were able to generate embryos with this knockout and then introduce stem cells from wild type mice that were labeled with GFP to see if they would occupy this niche here. This chimeric embryo, in turn, was then transferred to a pseudopregnant mouse, and then the offspring were studied. And what they found was that these offspring actually had a pancreas that was derived from the stem cells of the wild type mice. And so these studies now have led to, or this approach and these mouse-to-mouse chimeras have resulted in the generation of not only pancreas, but also kidney, lung, and forebrain. So the Nakaguchi group then asked the question of whether this could be done between species. To address this, then they took stem cells from rats and introduced them into the knockout embryo of these to generate this chimeric blastocyst. And again, transferred to a pseudopregnant mouse. And what they found is that they could generate a rat pancreas within a mouse. So this interspecies chimera now has been successful not only in the pancreas, but also in generating kidney. The Yamaguchi group in particular have shown that the islet cells that are generated from the pancreas could be transplanted and actually cure diabetes in animals and mice that have this disorder. So it shows the functional functionality of these transplanted cells. So to translate this clinically, we need animals that are larger in size, that have organs that are more similar to humans. And so the next step in this process was work also done by the Nakaguchi group in trying to generate a pig that would have a pancreas. So this is an animal that has organs that are very similar in size to humans. We're able to generate a PDX1 null animal by increasing Hess expression. And so these, the fibroblasts from these animals were then, the DNA was taken and introduced into the nucleus of an enucleated egg to generate a null embryo. And then that blastocyst in turn was injected with wild type stem cells. And what they found was that the transfer of this into a pseudopregnant sow resulted in pigs that had a pancreas that was derived from the donor stem cells. So this general approach then has resulted now in the production not only of pancreas, but also kidney, liver, and vasculature in these pig-to-pig chimeras. So obviously, what we want to do then is generate human organs within these large animals as biological incubators. Excuse me a second. So that could potentially be used for transplantation into humans. So this would require the generation of or the use of human pluripotent stem cells that are placed into the blastocysts of animals that have been genetically engineered to delete particular genes that are specific for the target organ of interest. And so this approach has been attempted, and so people now, the various groups have been able to generate cells that are human muscle cells, human neuronal cells, endothelial cells, and hematopoietic stem cells. But the contribution of these cells are actually quite limited. And so there's a hurdle here that we have to overcome in terms of sort of optimizing the cells, the human cells that are being used, or somehow manipulating the host embryo to make it more humanized. And so these are challenges that we need to confront. And there's the hurdle here in terms of funding by the NIH. So any proposal that's submitted to the NIH where that proposes to inject human stem cells into a blastocyst of any animal will not be funded. And so this concern is a result of a moratorium that where the NIH will not fund any of these studies, and because they wanted a period of where the public could comment on their attitudes towards this type of research. And so this moratorium was set up about 3 years ago. And since that period of time, there has been no report by the NIH as to the results of this moratorium, the results of the comments by the public. And so what we wanted to do then is to determine what are the attitudes of the American public in terms of generating human-animal chimeras. And so we began to sort of adopt a strategy that was used by a group in Japan to interrogate the attitude of the Japanese public in terms of human chimera, animal chimera research. And so the approach that was taken for this survey was to break up the research into 3 steps. The first step involved the transfer of human iPS cells into an embryo to create this chimeric embryo to really study the growth process. The next step was, for step 2, there was the transplant or the transfer of this embryo into a surrogate or a pseudopregnant sow, and then generating an offspring that had an organ that could then be transplanted. And the third step was the actual transplantation of this organ into human individuals. So we wanted to determine whether the American public would accept any or all of these steps in the process of this chimeric research. So we wanted to determine the number of respondents that felt that this research was totally unacceptable, or whether they felt that all steps were acceptable, or whether the first 2 steps in this process were acceptable, or only the first step in this process was acceptable. So when we analyzed the data, we're actually quite surprised. We found that over 80% of the respondents were actually could accept some aspect of this research. About 60% could accept all steps. About 10% of the respondents could accept the first 2 steps of this research, and then about 10% only the first step of this chimeric process. And less than 20% then felt that this research was totally unacceptable. We then went on to ask the question of whether the respondents would be willing to actually donate their own somatic cells to generate iPS cells for this research. And so what we found is that about 75% of the respondents were actually accepting of any of these processes, and about not quite 50% associated with accepting all steps. About 15% would accept the 2 steps, first 2 steps, and about 15% only the first step. And so about 25% found this to be totally unacceptable. We then wanted to ask the question, well, do you think that this research should be socially acceptable? And so what we found here is that now over 80% of the respondents felt that some aspect of the research would be socially acceptable to them, and that less than 20% felt that this research was totally unacceptable. We next addressed the question of individual organs. And so we wanted to see whether people felt that they could accept the different organs that were being generated, or whether they could not accept these. And so we looked at liver, heart, pancreas, blood, and skin. And what we found was that about 60 to 75% of all the respondents were actually found that to be acceptable to generate these particular organs. And about 25 to 30% felt that either they could not accept it, or they somewhat could not accept the generation of these particular organs. We looked at brain, however, and also germline cells. What we found is that there was a decrease in the number of people that would accept it. So with brain, there was about a 50-50 split between those who could accept it and those who could not. And with germline, we saw an even greater lack of acceptance on the part of the people who were producing germline cells. We then split this up and looked at the political views of the respondents. And so what we surveyed we asked whether they would categorize themselves as being very liberal, somewhat liberal, moderate, somewhat conservative, or very conservative. And what we found is we expect that those that were self-described as liberal or moderate, over 80% were in favor of generating a liver through this process, even those that were moderate, about 60%. Surprisingly, even those that had self-identified as being somewhat conservative or conservative, and even very conservative, had a very favorable response in terms of generating liver using this chimeric approach. The results are fairly similar for heart, except for those who were self-described as very conservative. We found that there were fewer of them that would accept this type of approach for generating a human heart. When we looked at blood, we found a very similar profile. Again, acceptance by all groups, and even those who were listed as conservative and very conservative. Same is true with that of skin. However, when it came to brain, what we found is that only the very liberal group was accepting, or had a high level of acceptance. The others were pretty much 50-50. They were split in terms of their acceptance of generating brain cells using the chimeric approach. And those who were very conservative actually could not accept this particular approach. And then the same was true of the germline cells in terms of the different groups. Then we looked at religious affiliation. And so we wanted to determine, so we asked whether they had any religious affiliation at all, or whether, and what we found is that about half of the responding group did have a strong religious affiliation, and then the other half did not. And what we found is that, as we expected, those that did not have a religious affiliation had a high level of acceptance of generating liver. And even those that had a religious affiliation, over 60% would accept generating a human liver in a chimera. That was true. The same profile was found in the heart, and also with blood and skin. And what we found, though, is that there was a change when it came to generating the brain, so that those who did not have a religious affiliation had about a 55% acceptance, and those that did have a religious affiliation dropped to 40% in terms of their acceptance rate. And the same was true in terms of germline. And so again, about a 50-50 split for those that did not have a religious affiliation, and this dropped down to about 25% for those who did have a strong religious affiliation. So in summary, I think that there's really a lot of advancement that's going on in terms of being able to generate whole organs in these chimeric animals. The majority of the U.S. public appears to be in favor of each of the steps of the chimera formation process. And even groups that self-identify as politically conservative or strongly religious support this general approach of producing human organs in these chimeras. And so we feel that the American public now is ready to accept this type of research, and that the NIH should reconsider the current moratorium and that revokes funding, or should revoke the current moratorium and then allow funding for this research to proceed. So with that, I want to acknowledge the Genesis Project group here at the University of Minnesota, and also with Carbonetics and the group at Kyoto. The people that are shown here in red are those that participated in formulating and carrying out the survey. So with that, I'm happy to answer any questions.